Good day and good evening. My name is Monica Farfan. I am the Executive Director of the Global Soil Biodiversity Initiative, and welcome to GSBI Speaks. We have a wonderful program for you today, but first I wanted to go through a few housekeeping details. First of all, the GSBI Speaks webinar series is going to be on hiatus for July and August. Please check our website for registration information for our September 2021 webinar. Today, I would like to also share with you that this is being recorded, and the recording will be available either later today or tomorrow. If you registered for this webinar, you will receive an email regarding that. Next, I would like to let you know that we will be taking questions throughout the webinar. However, only in the discussion portion of this webinar, so the last hour, will those be addressed. For those of you who would like to add a question to the queue, please use the Q&A box at the bottom of your screen. And now I would like to introduce our moderator today, Katie Suding, from the University of Colorado. Katie is known for her work in ecological restoration. And please, Katie, now the floor is yours.
Great. Thank you, Monica, and welcome everyone to our panel today on soil biodiversity and ecosystem restoration. So as Monica said, my name is Katie Suding. I'm a professor at University of Colorado, and I will be moderating this next hour and a half. And I want to start out just by pointing out some really exciting news, and I hope everyone was aware of it, but this Saturday, the UN chief, Antonio Guterres, kicked off the UN Decade of Ecosystem Restoration. And in his speech, he pointed out that it's the next 10 years that are our final chance to avert climate catastrophe, turn back a deadly tide of pollution, and end species loss. So they are putting a huge emphasis on the next 10 years and on ecosystem restoration. So we are starting the decade of ecosystem restoration. So this panel is super timely. And alongside forests and oceans and deserts, soils are really taking a center stage. For instance, the UN Convention on Combating Desertification has set the ambitious target to restore 100 million hectares of degraded land and soil capture around 250 million tons of atmospheric carbon in the next 10 years. So yeah, this is big, and it all, at least in my opinion, revolves around soil. And why I think everyone is really super excited about this ambition. You know, it's a little nerve-wracking as well. And I just want to start with a little anecdote about my personal work. So when I have looked into issues of restoration time and time again, I found that when a system is stuck, when it's really seems difficult to guide recovery, and we're pulling our hair out about what to do, you know, the key component almost always, at least in my case, is the soil community. And it rules many, if not all, of the plant recovery and ecosystem dynamics in these semi-arid grasslands and prairie systems that I work with. So it really is key. And so I think it is very much a key consideration in these next 10 years if we are going to meet these ambitious goals. But we also often hit the wall once we pinpoint that it's the soil communities that are key to recovery and ecosystem dynamics. That what are we going to do? What are we going to do to address this limitation and restore soil biodiversity? Get the important symbionts back into the system. And this isn't really as simple as, for instance, just planting a tree. It is a super complex problem. And in many cases, I've walked away scratching my head and not being able to provide great solutions to the managers and policy makers that I'm working with. For instance, just one example of this, the native grasses in California can persist and resist invasion really well, and with a lot of environmental threats and pollution, drought, so on. But when they're removed and we try to get them back, so try to restore them in a system, we really have very little luck because the symbionts, the microbial symbionts in the soil are not there. And we try time and time again, we try different things, try all these ways of trying to restore the soil community. And it is very, very difficult to get them back in the system. So the fabulous thing about where we are at in this research about soil biodiversity and restoration is that we're learning so much about these linkages between the soil community and recovery and restoration, as well as we're breaking down these walls about how to use that knowledge and really apply it to restoration actions. So there's a lot of exciting breakthroughs right now. So right now I'm super excited to hear from 4 of my favorite soil restoration scientists, and together talk about soil, restoring the soil, the role of millions of microbial and animal species that live in soils, in restoring an entire ecosystem. So we are so fortunate to have these 4 scientists with us today. And so I'm going to briefly introduce each one, and then I'm going to ask them to give a short introduction for the first bit of our session today. So we are fortunate to have Sara Baer from the University of Kansas. So Sara, Susana Rodríguez Echeverría from University Coimbra, Portugal. Susana?
Hello.
We have Jasper Wubs from ETH Zurich.
Hi, everyone.
And lastly, we have Raul Ochoa from University of Cadiz in Spain. Raul. We have asked all panelists to talk about their amazing work on the subject. They bring a huge diversity of perspectives and expertise, as well as looking forward about our major gaps and understanding what can be done and how we can move forward, particularly in these next 10 years. We will hear from each, and we're not going to stop for questions in between, and then we'll open it up for questions as a group. So please type them in the question answer section of the Zoom, and I'll try to get to as many as possible. So I'm pleased to turn this over to Sara for her to talk about her research. Thank you, Sara.
So it should start soon. Okay, well, thanks for inviting me to present on this at this very timely forum on soil biodiversity and ecological restoration. Much of my research has focused on the role of plant communities and soil properties as drivers in reversing ecosystem disservices caused by agriculture. So when grassland is converted to row crop agriculture, numerous disservices result. When the soil is tilled, it disrupts the soil and reduces soil carbon by promoting erosion and exposing carbon that was protected in soil aggregates to decomposition. And this results in a release of that carbon as carbon dioxide to the atmosphere. Furthermore, when we fertilize these soils, there's an increase in nitrogen export from the system, and fertilization also promotes the production of nitrous oxide from soil, which is a more potent greenhouse gas than carbon dioxide. So today I'm excited to share with you some of our discoveries on the role of soil texture and managing for plant diversity on reversing these ecosystem disservices. And I'll talk about knowledge gaps that we've learned about the role of soil biota in restoring ecosystem services that are relevant to mitigating climate change, and some implications for land policy. So my colleagues and I have studied the role of soil texture on soil recovery in Nebraska. Here we used many grasslands that were restored with native warm-season grasses, but existed on two very different soil types. So within each soil type, we sampled many fields that were restored for different periods of time in order to model below ground recovery. And what we found was recovery in many aspects of the soil microbial community. Recovery in the number of phospholipid fatty acid biomarkers that are extremely coarse measure of microbial diversity, recovery of microbial biomass in the soil, the ratio of fungal to bacterial biomass in the soil, and the recovery of arbuscular mycorrhizae fungi biomass in the soil, but all of these patterns only occurring in the clay soil and not in the sandy soil. And so those soil biota are required in the soil and their recovery to form these aggregates that ultimately protect carbon in soil. And so what we saw was only an increase in recovery in this aggregate structure happening in the clay soil. And consequently, we only observed an accrual of carbon occurring in these clay soils. We used a different pair of restored grassland chronosequences to look at the role of managing for plant diversity on soil recovery as well. And here we did this in Illinois, where we used, again, a sequence of fields restored for different ages that were restored with many species and managed frequently with fire, and compared that sequence to fields that were restored with far fewer species and managed with infrequent fire. And so results from this study showed that managing for diversity results in a continuous increase in root biomass, a more predictable recovery of the active microbial biomass, and a faster recovery of that soil aggregate structure. Managing for diversity also reduced the amount of inorganic nitrogen available to be lost from the ecosystem through leaching, and less potential nitrous oxide flux from soil when managed for high diversity. So when we look across numerous studies that have quantified carbon accrual rates in cropland restored with warm season grasses, we see large variation in the recovery rates of carbon. And so this indicates that we don't know all of the factors that are contributing to the capacity for which they can accrue carbon. And other questions emerge, like how will the capacity for restored grasslands to sequester carbon change as climate changes? For example, how will drought affect the recovery of soil biota and their critical role in forming those aggregates that protect carbon? Furthermore, we don't know how tightly coupled the soil biota are to plant materials that are used to restore grasslands. In other words, will we need to use co-assisted migration of plant ecotypes and soil biota to fully restore services as climate changes. So these knowledge gaps are relevant to land policy. So the United States Department of Agriculture's Conservation Reserve Program is the largest incentive and opportunity to restore grassland on former agricultural land. The USDA plans to enroll over 1 million hectares with a targeted focus to help mitigate climate change. Our research shows that the capacity of restored grasslands to accrue carbon depends on soil texture, and likely many other physical factors and feedbacks between plants and soil. Moreover, there are other ecosystem services that restored grasslands perform and could potentially perform better if managed for high plant diversity.
Thank you, Sarah. Amazing. Now I'd like to invite Susana to share her screen and talk about her work.
Hello, everyone. Hi, Katie. Thank you. Thank you for the invitation. I'm going to talk a little bit, if this thing wants to work. I'm going to talk about a specific group of, a specific type of organisms in the soil. I'm going to talk about mutualists, about symbiotic mutualists. These are microbial bacteria or fungi that live very closely ligated to plant roots. We can divide it in two main groups. One of them is the symbiotic nitrogen fixing bacteria, and the other one will be the mycorrhizal fungi. The symbiotic nitrogen fixing bacteria are well known for their association with legumes. About 17,000 species of legumes associated with this bacteria. And mycorrhizal fungi is probably one of the most ubiquitous mutualisms in nature. Many people that works with mycorrhiza will say that most plants don't have roots, but they have mycorrhizas, since about 90% of the studied plant species form this kind of mutualism. So they are very important for plants, for terrestrial ecosystems. They are mutualists that are involved in nutrient uptake. In the case of the bacteria, they allow plants to use atmospheric nitrogen. So they can fix this atmospheric nitrogen into organic forms that can be used by the plant. And thus they have a competitive advantage in nitrogen poor soils, such as many soils that are degraded by anthropogenic actions. The mycorrhizal fungi increase plant uptake of different nutrients, mainly nitrogen, phosphorus, but also micronutrients, and also water, which is very important in many arid and semi-arid ecosystems and for the future due to global change. They also protect plants against pests, and so they promote plant growth and reproduction, and they are very important determinants of plant communities, diversity and productivity, and for the stability of soil aggregates, as Sarah was talking about. So as you can see, these are very important for terrestrial communities functioning and diversity. And when a disturbance happens, we need to think about them in our plans of restoration. As for other projects of restoration, we can approach this as a passive restoration or an active restoration. So the simple thing that we can do is just stop the disturbance and wait for natural processes to happen, for natural recolonization of these soils by these mutualists from nearby conserved areas or ecosystems. It's a slower process, but it's also very cheap. And since it's the natural succession, the success of this approach is guaranteed because it will happen what it has to happen in the ecosystem. That it doesn't mean that it's going to recover to pristine conditions, but it's what it can be done. Now, in many cases, we don't want that. We want something quicker or a stronger action. And then many types of inoculations have been tested for the use of mycorrhizal fungi or nitrogen fixing bacteria in revegetation projects or restoration projects. It is the best strategy for heavily degraded soils, and there are many studies done, especially for soils contaminated with heavy metals or oil residuals. But the success of these projects, of these active restoration inoculation projects, depends on the identity of the plants and the mutualists, but also on the requirements of plants and the conditions of soil, as Sara was saying before, and also of the inocula, as we will see later. As an example of passive restoration or natural recolonization, there are many studies that assess the diversity of a kind of mycorrhizal fungi that are associated with temperate forest trees. We have been doing some research on this in the last years, specifically with beech trees, with Fagus sylvatica, and with the associated mycorrhizal fungi, which are essential for the growth and physiology of these trees in natural forests. We studied forests affected by two different types of disturbances. In one case it was mining that destroyed the forest, and in the other case it was in the conversion of forest to pasture land during the 19th century. In both cases, these disturbances stopped a while ago. In the case of mining, it was between 100 and 140 years ago. And in the case of pasture land, they were abandoned about 60 years ago. And in both cases, these are different sites in the north of the Iberian Peninsula. In both cases, the forest regenerate naturally. We studied what happened with the communities of mycorrhizal fungi associated to these species. As you can see in this image, you have here a mine which has been recolonized by the beech trees, the associated soil organisms. And here you have a photograph, aerial photographs of the forests that were cleared for pasture land and how they have regrown in the last 50, 60 years. In both cases, we have diverse and abundant community of ectomycorrhizal fungi associated to the roots of these trees. But as you can see in both cases, after 100 years or 60 years of recovery, the communities associated with these new forests are different from the communities associated with the forests that did not suffer the perturbation. So the recovery of the mutualism happens in a natural way, but we don't know if these differences in community composition are having any effect on the functioning of the forest. And this is one of the things that is starting to be addressed now. Also, the colonization will depend on the conditions of the soil, that sometimes, in most cases after disturbance, the concentration of nutrients and the pH of the soils is different, and that is not going to allow the establishment of all of the same species that were there before. But also, it will depend on the capacity of dispersal of these fungi from nearby forest. The other option, the inoculation, is management option that is very well established and that has been using a lot. Also because inoculation with nitrogen fixing bacteria or mycorrhiza is something that has been very well established and studied in agronomy and also in the gardening industry. You can, if you look up in the net, you can find lots of different inoculants to apply for your plants or your crops. But what different studies have shown is that you shouldn't use these commercial inoculants in revegetation projects, in restoration, because they contain foreign species that can be potentially invasive. And also, in many cases, they are not successful in natural terrestrial ecosystems. This meta-analysis shows precisely this. You can see that the success of the restoration project improves with the application of mycorrhizal fungi, but this improvement does not happen for commercial inocula. So the best option is always to obtain an inocula from reference ecosystems that are nearby the disturbed site. And you can use a mix of isolates from these reference ecosystems or single species isolated from these ecosystems. Or you can use the whole soil as topsoil transplants, or cultivate these mutualist appropriate nurse plants and put these to help restoring the system. There are many different options. In many cases, they have to be tested, and because there is no one only solution for all cases. One interesting case is the restoration of grasslands, especially in North America. There has been many studies done in restoration of grasslands and the application of mycorrhizal fungi in this restoration. The inoculation with local native fungi increases plant survival, richness, and productivity in these projects, and promotes the establishment of late successional plant species, which are more dependent on mycorrhizal fungi. So they are essential components of the ecosystem and essential components for the recuperation of this ecosystem. Here I just saw very quickly a piece of a study that we did in the recovery of coastal sand dunes that were invaded by an Australian acacia, which forms very dense stands and changes completely the characteristics of soil. The soils are very much enriched in organic matter and nitrogen. We did a pilot study using native shrubs, native legume shrubs, to recover the vegetated areas. And we also saw that in the nodulation, so the symbiosis with the nitrogen fixing bacteria will increase the probability of survival and the growth of these plants. So again, as for annuals or perennial annual species, you can also use this kind of inoculation techniques with native mutualists to improve this, the success of restoration projects using woody species. So there are many, if you go to the literature to do a search, there are many studies trying to do this. Talking about the major gaps in this area, although there are lots of research, there are still many gaps in our understanding of how this works in different ecosystems, because most of the studies are done in temperate ecosystems, and also many of these studies are done only with one kind of mutualism, and it was very interesting to use both nitrogen fixing bacteria and mycorrhiza when legumes are present. Also, many of these research reports are focused in individual plant growth. We need to expand our knowledge on different ecosystem services and different processes to see if the inoculation or the natural colonization are better options for the recovery of this ecosystem. Thank you very much.
Thank you, Susana. Yeah, that's very close to my heart. That's just incredible work. So now I'd like to invite Jasper to talk about his work, and I'd like to just remind the audience, please, if any questions occur to you, just please use the question and answer tab to ask them whenever you have any. So thank you. Okay, thanks, Jasper.
Thanks, Katie. I apologize for the black boxes in the top. I cannot get rid of them. So indeed, my name is Jasper Wubs. I work at ETH Zurich, and my work focuses on steering soil microbiomes. And I came to this as a plant ecologist, focusing on plants. And I was introduced to the beautiful world of soil biota by Martijn Beesem and Wim van der Putten during my PhD. And they showed me the amount of interactions that were going on above ground and below ground. So above ground, you have 30, maybe 40 species per meter square. In the soil, you have thousands of species that live together, bacteria, fungi, all kinds of fauna, and all of these interact in some way. And so I think, you know, there have been several decades of research on this, and I think the field is moving into the direction where we're looking into ways in which we can manage or manipulate these interactions for specific goals, for instance, restoration. And my research starts from a different point than most people. My research assumes that there's not one or a few microbial players that are super well functioning, that do everything. But in fact, I think, or I start from the point that actually you need to get a whole community of soil biota to move in a direction that's beneficial for what you want to do. So I'm looking for methods to change and steer the whole soil microbiome for soil functioning in general. And I've used different methods to do that. One is soil inoculation that I've used in field experiments, where we could show that the microbiome can actually determine in which direction the vegetation is developing. And the other method is host-mediated selection. So there's a way to manipulate the composition of the microbiome, and we use that now in central Kenya to see if we could optimize maize cowpea rotations that are dominantly used there on tropical soils. Now, for the future research, there's three things that I would like to highlight. One, I think is very well known among soil scientists, soil ecologists, is, okay, we have been doing a lot of research, but most of it's been done in pots and in the laboratory. We need to take this outside, and we need to adopt methods that allow us to give credible answers to these pressing societal questions, particularly with respect to restoration. And we need to do that in the field. And we also need to link, I think, much of the soil biodiversity more explicitly to how soils function to be able to give those answers. Now, the second is, how do we maintain the potential soil biodiversity? I was recently involved in the State of Knowledge report by the FAO. And there's a lot of microbes that have been found that they're beneficial for bioremediation, for instance. But when you look at those papers, there's actually very few that document where these microbes come from. So it seems that the best guess we can give that if you want to, you know, if you want to get rid of an oil spill, for instance, you go and look for microbes that live in oil spills. And that's about the best answer I think I could give you at the moment. So I think we need to dig into that deeper. Where is this biodiversity coming from? And also, how can we maintain it? Do we need to maintain it somewhere, you know, outside of the soil in a laboratory, or can we in the management of our landscapes, can we make sure that we keep that biodiversity there? And the third thing is a bit more a broad scope thing. It's about the ethics and sociology of microbiome tweaking. So I think many people at least I in restoration ecology, we have a bit of a technocratic approach to things. We find ways to fix perceived problems. We also have to take into account how society and how people relate to these innovations or these interventions that we propose. We should discuss what is proper in moral terms and in sociological terms in different cultural contexts. And one thing that has always struck me is a discussion with a philosopher. He said, well, okay, the better you get at restoring nature, the more ammunition you're actually giving other parties to destroy nature, because you're now better able to restore it. So why bother about conservation? Why do you need to conserve? And I think in all our restoration efforts, we are seeing that we can restore, but we cannot restore perfectly, not even close to perfectly. And so I think there's a real risk that we should not overemphasize our ability to restore. We can restore, we can make things better. But it's not nearly as good as what we're losing, and so I think we need to be explicit about that and study how that relates to society generally. Now for policy, I think indeed this link to society is very much key. We need to find out not only how people in social sciences are doing things, but particularly we need to find out what is relevant for local communities, what questions are relevant for them, and what are bottlenecks in the problems that they perceive, and can we find solutions for that? And I think in the resource-rich communities that I, for instance, work in, we need to develop the technologies. We need to develop the options of improving things, and those can then, after we know the rules of the game, for those interventions, we can start applying, and we can do more direct research in other communities and get them to solve the problems they may be in. That's all I wanted to say. Thank you.
Thank you, Jasper. And I really appreciate you bringing up the social and policy dimensions of what we're talking about today. That's great. Raul, you're next.
Okay.
I'm going to hand it over.
Okay, perfect. Thank you. Okay, I will share my screen now. Okay, I hope you're seeing it. Okay, so I would say, when you look up and contemplate the night sky, what do you see? I personally see order. Now, take any living organism or a part of it, like, for example, these grapes. What do you see now? I see order too. Actually, nature is ordered across scales, and this order means that everything must be somehow connected to everything. Such orderly connections define how systems are coupled. This order arises naturally, which means that it is self-organized. The other two main characteristics of self-organized ordered systems is that they are self-sustaining and dynamic, or in other words, they evolve. The amount of order of a given system defines the energy stored within that system. Hence, orderly coupled ecosystems should be able to function more efficiently. In our planet, the energy comes from the sun and from the interior of the earth. And we know that soils play a key role in the processing, transfer, and storage of that energy. Somehow it is like the soil is the battery that helps to keep ourselves and the earth ecosystems running. This is actually compatible with the view that soils are central to support life on Earth. And of course, soil biodiversity is central to this. Now, I hope that I have convinced you that order across scales, or in other words, coupling, is a critical feature of functional ecosystems, including production systems. Actually, based on associations in absolute value and a null modeling approach, which is what I use to define coupling operationally in my research, ecosystems can be found in three coupling states: coupled, decoupled, and anti-coupled. We ecologists normally look for coupled patterns, right? Seldomly care about the coupled ones, and I would dare to say that we never look for anti-coupled patterns. Am I right? Like I said, nature is ordered across scales. So what happens when we break the fractal nature of coupling? The disorder wave propagates to the lower levels, and that results in a generalized energetic downgrading of ecosystems. So in other words, if we want to bring back the energy and the functioning of the soil battery and its associated biodiversity, we need to bring the coupling back. We must do so by considering the three main characteristics of coupled systems that you know already: that they are self-organizing, self-sustaining, and that they evolve. And also that they need to be fed an ordered source of energy to be able to comply with the second law of thermodynamics. And so how can we bring the coupling back? In my opinion, we need three basic ingredients. First, we need to bring back the soil organisms that may be no longer present. For example, through inoculation, like Jasper or Susana said before. We also need seeds of plants that will contribute to recharge the soil battery by pumping energy from the sun into the soil, like Sara, for example, suggested. And last but not least, we need large animals that are key to the circular economy of the system. After this, everything else should follow. And this is actually the approach that I'm taking in my research to regenerate the life of vineyards from the Red Sherry region in the south of Spain. These vineyards have a hugely long history since the times of the Phoenicians and the Romans, but they are now highly eroded and biologically impoverished due to intensive management. My goal with my research is to bring the coupling back. For this, I'm leading two research and innovation projects. One is called Living Soils and the other one is Future Vines. One of my main goals with these projects is to generate manuals that can be available to growers and winemakers and to be in touch with various stakeholders, thus being able to steer the future sustainability of vineyard soils from my region. To me, one of the critical aspects to achieve this is to create different types of gradients that will channel the fluxes of energy and matter. If you think about it, this is somehow equivalent to bringing back color to ecosystems. So let's put back the color to our ecosystems, and let's not forget that soil organisms are a critical component of the unseen palette. And with this, I thank you for your attention.
Great. Thank you, Raul. So now I'd like to invite all the panelists to turn on their videos, and we're getting a lot of questions, and I'd actually like to start with one myself, and then I'll try to get to as many of these questions as I can. They're just amazing thoughts coming from the audience. So what I am really interested to know from you four is, just given where we are now and the amazing progress that's being made, and all the information that you're sharing with us, where do you think we will be in 10 years? Or where would you like us to be in 10 years in terms of kind of incorporating soil biodiversity and soil processes in these in grand kind of restoration challenges? Take it, Sara.
So, I'm hopeful that, I come from the grassland perspective, and agriculture is a major degrading influence on soil. But I think the more extreme climate events that we have will make producers, or I'm hopeful that it will result in a feeling of needing to be a better steward of land in regions that are resource scarce. Growers are knowledgeable of soil biota and aggregates and mycorrhizae because the resources are so limited. So I actually think it might be sort of forced upon us collectively through nature, and that force, I am hopeful, will result in a greater trend of using conservation practices in agriculture and conversion of a lot of marginal lands to grasslands. And I hope that this decade of focus on ecological restoration will result in more incentives to do that.
Awesome. Someone else?
I mean, in my opinion, I would say that it's important to consider that somehow the energy is within, right? So there's no alternative to using the power of nature to drive the functioning of ecosystem. And the same happens if we want to regenerate. So I'm pretty positive that we will all realize that is it's a universal truth. And many of us know that already. Is like our health depends on ourselves somehow, and how we manage our own lives. And the same happens with ecosystems. So once we all realize that, I think that the solution is at hand.
Jasper, I think you were gonna, yeah.
Yeah, sure. Yeah, I really love the points of Sarah and Raul. I want to add to that a bit. I think there's a lot of momentum growing in different parts of society, and I guess in society at large, about people realizing the value of biodiversity, of soil biodiversity, and of the need to restore ecosystems. So I'm very hopeful that indeed this decade will bring a change. But I also think that we as scientists need to work hard to make sure that we bring all the evidence and all the data together, because it seems that in many cases blanket solutions do not work everywhere. So we need to untangle what is working where and why by bringing all that information together. And also bringing the socioeconomic context into it. And I think that will be a major step for us to do in the coming years.
Excellent. I agree. Yes, Susana? Yeah.
Yes, I just wanted to add to what Sarah just said, that I think is very interesting. I think that if we look from now to 10 years, we look from now to 10 years backward, in the last 10 years, there has been a very, the interest for soil and soil diversity has grown a lot in the community of ecologists and restoration practices and also managers. And I think that is not going to stop, it is growing. And I agree with Jasper that we need to make an effort to transmit all this information in a better way, and to say that not, like he was saying, that we don't have a unique solution for everything. I think that is very important, because when something is tried in one side or two sides and people doesn't see a result, then it's very difficult to recover that confidence. So it's better to go with the information at first hand, to try to keep people interested in what we're doing and in how things can be done. And also, I think that many people is now, at least in Europe, or many people that is in close contact with land, like small agricultures or small farmers, are more interested in how the soil works, and bringing that knowledge into management is a very interesting thing that is going to happen in the next years. Thank you.
Great. Yeah, I think just paying attention to land is incredibly important. So there's been a lot of questions from our audience about kind of active interventions to address mostly soil microbial diversity and processes. And so I just want to ask a couple of questions from Thomas and Francisco, in particularly about kind of this active restoration. So first question is just one really general, is there a way to kind of address, if you want to do some active restoration to address soil biodiversity, which groups would you address first? And how would you go about doing that? Like, is there particular methods that you think are particularly promising? And I know, Susana, I think we should probably start with you first, because you kind of talked about that most explicitly in your presentation, but I'm sure the rest of you all too have thoughts about that too. So.
Well, I mean, from the point of view, of course, I will say that you need to restore the mutualists together with the plants, because it's going to be very difficult. If the soil is very degraded, it's going to be very difficult for most plants to establish and grow and reproduce if they don't have their mutualist there. But also, and you can try to do that by inoculating isolates obtained from nearby ecosystems, like more reference ecosystems. But also what you can do is to use topsoil transplants from nearby ecosystems, and then you bring the whole soil community together, and that will work much better, because otherwise it's going to be very difficult to restore all the functions of the soil. But if you have, again, is very dependent on the situation and which is the perturbance, the disturbance that happened in the soil, and the current status of the soil. If your soil is totally degraded in physical terms, then you need to address that before you introduce anything.
Great. Thank you, Susana. Anyone else have any ideas about how to approach active restoration to address the soil microbial community?
Yeah, I don't know. It seems hard to give very general rules. I mean, if indeed the soil is physically destroyed, you have hardly any microbial biomass, then restoring that basal biomass, the bacteria, the fungi, seems like a good first step. But of course, you need active root exudation to keep these guys going as well. So you need to introduce the plants, I guess, at the same time. There are more specific things that you could do, I guess, if your problems are not so big. If you have a problem with compaction or so, maybe you could do things with earthworms or stuff like that. But I think it really depends on the type of degradation and where you're at, what should be the focus.
Raul?
Yeah, so to me, one critical aspect would be to restore the aerobic decomposers, and particularly aerobic decomposers that simultaneously support the immune system of the plants. For example, many of these aerobic decomposers can also simultaneously produce vitamins and other components that are critical to the health of the plant. So if I have to make a guess or to propose something, I would say let's go for phyla like actinobacteria and things like that. To me, those are critical to get the functioning, started.
Great, yeah. Sarah, do you have anything to add, or?
Yeah, I wasn't sure if we were going one by one.
No, don't. Just jump in.
A lot of our work shows that there is a lot of recovery that does occur naturally. And I think imparting as much plant diversity in this system will help the below ground diversity. But I think the answer to that question, at least in prairie systems and systems that I work in, are forthcoming. We have a project going on here in Kansas researcher at the Biological Survey has taken like blocks of native prairie, undisturbed prairie that they have removed to build roads, and then inoculated those blocks into an agricultural field to look at that dispersal of the soil microbial community, fungi and bacteria. And then certainly there's a lot of research on very, very host-specific mycorrhizae that do need to be cultured and need to be cultured on their living host plants, and then using those inocula or those potential plants. So I don't think it's easy. You know, I think there's natural recovery, but if you really want to rapidly restore soil microbial communities and diversities, it would take a lot of effort.
And so I guess just as a synopsis, I think that the idea is about kind of whole soil restoration and kind of trying to introduce the whole suite of soil biodiversity back in the system, along with the other components that the soils, coupled with particularly plants, are probably essential. I have two really more specific questions that I think might be just helpful to address really quickly. First of all, there's a question about if you inoculate a system, how fast would you expect that inoculation to spread, you know, in terms of affecting the whole community, and whether that's a quick process or whether that's a slow process. Jasper?
Yeah, I haven't done really active research on that, but just from observations, it seems to be going very slow and rather limited. So if you inoculate a patch, it seems to pretty much stay there. It does spread a bit. I mean, but we're talking meters at most, I would say, for many things. I mean, there are papers that show that on a meter scale, you know, if you inoculate a mycorrhizae here, then the plant 2 meters away can respond to that, does respond to that positively. But I don't think it will be hundreds of meters in a few years.
Right. Sarah, you agree? Yeah, I think that inoculation is something that, even though a lot of microbial processes are fairly quick related to plants, the spread of an inoculation is not necessarily lightning fast. And there's a lot of interactions there. I think that gets to Raul's comments about coupling and decoupling, right? You have a system that's in place that could be degraded, but could be really resilient to the interventions that you're trying to apply. So I wouldn't expect necessarily that process is quick or easy, even if you add the right or the organisms that you desire. Yeah, Jasper?
Yeah, just one thing to add maybe, and this is something I wonder. I mean, a lot of the research on this seems to be done in flat places. So the Netherlands obviously qualifies as a flat place, the Midwest in the US as well. And there, I guess most of the water flow is vertical down the profile. So I wonder if you're on more sloping terrain, if the spread rates going down are better. But I don't know if that's the case. But I think it seems to me that there's a mass action effect. You know, you need sufficient numbers of microbes to really transition a system, and this doesn't seem to be happening in a flat place, you know, going horizontally. But maybe if the water flow is more lateral on average, maybe that would help. I don't know if others have thoughts on this.
Sarah, do you have any thoughts about that?
So I often wonder if the stage of restoration matters. You know, is inoculating at the onset when you have, you know, no aggregate structure in a severely degraded situation. You might have high levels of nutrients, you know, extreme wet, dry conditions. I don't think we have a good sense of you know, when is the best time really to intervene? If we can restore some of the plants and some of the function, and let the ecosystem and facilitate the recovery, you know, when would be the best time to inoculate the soils? I just don't think, I don't know if we know. I don't know if any of you have seen any research on that, but we have a tendency to want to go in with immediacy and see grand change over short time scales. And that might not be realistic. And I want to give a shout out to Jasper, because his sort of the philosophy that we can repair everything can be dangerous, you know, because it can undermine conservation efforts. And I smiled because when I teach my restoration ecology class, that is the paper we open up with reading.
Yeah, I fully agree with that. I think rule number one in restoration ecology should be keep what you have. You know, try to stick with the ecosystems that are still looking okay. Try to maintain those. That is the best we're getting at the moment.
Right, and have humility, right? That you can't
Mhm.
you can't drive everything. Raul, I wondered if you could just jump in a little bit about thinking about these processes, and how would you take a system that is decoupled or maybe even anti-coupled, and kind of what does it take to kind of recouple those components in your view?
Okay, so to me, the first thing that I would like to say is that I consider regenerating ecosystems somehow similar to playing guitar, if you allow me the comparison, because if you want to play a tune, normally you want to, you know, touch different strings simultaneously. So playing a tune involves different strings, so playing a chord. And if we want to play, or let's say to reattune back that ecosystem, we need to consider that. And so recoupling means inoculating, of course, but that would be one string. Then we also need, like I was saying in my presentation, we need to create gradients. I think that's the core of how to bring back the function into ecosystems, because gradients is the only thing that allows energy and matter to flow. And that's what we want to recover. Actually, we want to recover functioning, no? And that function is only allowed if we have gradients. Otherwise, nothing moves. And if we create gradients artificially, for example, inoculating is creating gradients. Also, sowing plants is creating gradients of plant diversity. Obviously, that's not going to be homogeneous. And maybe along with those microbes and with those plants, perhaps you could add small amounts of clay, for example, in the form of pellets, and then you spread it over your fields, and then somehow you bring the large animals, which in my vision are like composters. So they're just recyclers, and we need to bring that back. And once we bring that back, we start the ecosystem. Let's say we awake the inner power of the ecosystems, although I know it sounds a bit esoteric, no, but that would be my vision. So I don't say that we need to leave nature to do its thing by itself. We need to help nature to awake by playing the strings that we want. In other words, we need to use the right chords. We need to resonate with nature.
Excellent. And I'm glad that you you brought up also the other ways of intervention, and particularly we have a couple questions about grazing or large animals or even soil fauna. So I was wondering if people could speak to what role you think those kind of other elements of the ecosystem have on restoring soil biodiversity or establishing those couplings. Sara, do you want to just talk about grazing?
Yeah, I think Raul brings up a really good point about the potential for bringing in or access to large animals, grazing animals. I mean, even proximity, I think, to other natural lands and connectivity could be important. I think there have been some earthworm additions to reclaimed mined lands, studies on that. I mean, but that's a lot. I mean, I know that they have been used as like a, you know, a bioturbation like tool to reduce compacted soils that then enable, you know, seeds to germinate and facilitate the cycling of nutrients. And of course, there are microbial hotspots as well.
Anyone else want to jump in?
I know very little about soil fauna, but I guess it could be important. I mean, I have heard stories about grasslands where, for some reason, the dead fungal biomass was not degraded, and I think I remember that was partly because indeed the shredders in the system, so the invertebrates that break down the massive chunks, they were not there, and so the whole decomposition by fungi and bacteria was not happening. And so it may be that in particular cases the system gets stuck, and you need these fauna to unstuck it. I have the feeling that more generally it's more about process rates than things being possible or happening versus not happening. So in many of these fauna, I think, speed things up, process rates, biogeochemical process rates. But you know, I guess it depends a bit on what you want from the system, whether that's beneficial, yes or no.
Great, thanks. I'd like to shift a little bit. We have multiple questions about monitoring and whether you think that we should start monitoring soil diversity and restoration to determine success, for instance? And if so, are there kind of keystone kind of indicator species that are taxa that might be ones to focus on? You know, whether we should start thinking about that as even an indicator of degradation, right? When a system is starting to lose its integrity.
I would say my first thought is monitoring soil microbial communities would not be easy. I mean, I guess I'm not exactly sure that, you know, the context of the question, like if it's something an agency could go perform easily. I mean, there are coarse indicators. I mean, looking at that fungal biomass could be really important. I mean, the fungi are critical in forming aggregates, and they're hyphae in the soil for transferring nutrients to plants. I mean, there are an aggregate stability. I think there are a variety of other metrics that might be a little easier to look at that could be indicative. So I guess what we really need might be an index, you know, an easily measurable index of the soil microbial community, and then that related to plants and diversity.
Right. I think that we shouldn't, you know, molecular methods are great and good for some questions, but certainly not for getting these practices to scale up on the ground to impact large portions of the globe. Anyone else? Yeah, Raul?
I mean, I would say that a very classic method to monitor the health of soils is to use soil enzymes, right? I mean, that wouldn't be soil biodiversity, but they are, let's say, majoritarily produced by soil biota. No? So perhaps if we want to monitor how soil biodiversity is doing, we may do it better by looking at how they are functioning. So in other words, we just may be able to measure enzymes, perhaps variety of enzymes and then derive an index. That could be a cheap alternative.
Susana, do you have any thoughts about root endophytes or that aspect of the system?
Yeah, well, it's like you were saying before, monitoring soil microbial diversity is expensive and not easy for managers, even for many scientists. I think that it would be better to focus on function, like they were saying, in soil functioning, not only enzymes, but other indicators of the composition or nutrient flows. And then for the root endophytes, there are cheap ways to analyze them, like from using microscopes or just for symbiotic bacteria, just to count the nodules, but they are all destructive measurements. So it can be something that can be done at certain points of time to check how it is going. But I wouldn't think that is going to be the best way to do it. I think it's better to analyze how the plants are doing. If you can detect any nutrient deficiency or any growth impairment or something like that, I think that plant monitoring is easier and will let you know what is happening with the root endophytes and the mutualisms. And also, that brings me back to one of the questions before about how inoculants would establish and disperse. I think usually you want to do the inoculation at the beginning, so you help the plants to establish, and then that will have that usually has a very good fast result on the plants. But it is true that there are not many monitoring schemes after that, after many months or years have passed, to see what happens. So maybe the inoculants are only there for the first initial stages and help plants, and then the system takes over and they disappear, and then we have to measure something different, something more functional.
I think a lot also about if you're in a degraded system, there are plant microbe relationships that exist, right? And so you have to almost knock out some like less beneficial relationships to establish the more beneficial, and that's pretty difficult to do, and it's not quite clear who has control, right? Whether it's the microbes or the plants, or I mean, well, it's probably not one or the other, right? So it's a very much just a tight interaction there to even. So just introducing the important players might not mean reestablishing the important players, and so I think that's also kind of a key question. There's questions about whether what do you think about biochar, compost amendments, and then also compost extracts to introduce or to try to address some soil processes. Anyone has any thoughts about any of those three techniques?
Well, I think there's evidence that adding the biochar, or that black carbon, can help with carbon accrual, in that it's a very difficult to decompose, but then organic matter can interact, with that substrate and sort of be adsorbed to it. It's like a physical chemical mechanism of carbon sequestration. So that's an option. And as far as, adding amendments, we've added carbon and sugar to some agricultural soils at an onset of a restoration and a larger heterogeneity experiment. And we found that can rapidly reduce nitrogen availability. So if high nutrients are a problem or concern, those organic matter amendments can be added, and they basically feed the microbes. The microbial biomass grows, and that microbial biomass immobilizes the nitrogen. So it can be a means to help mitigate maybe some excess nitrogen in a system if you want to restore, you know, plants that do better in low nitrogen environments, or reduce weeds.
Yes, yeah, absolutely. Hey, Raul?
So I would say that you could use compost in two different ways, or perhaps complementary ways. One would be to increase the carbon and the nutrients in the soil, or perhaps also to, let's say, to improve the soil structure. But another way to use compost would be just as a vehicle for microbes. So those are two different, perhaps, like I was saying, alternatives. So maybe, I mean, it's like when you take probiotics, sometimes you take them with the prebiotics, right? That help those microbes that you are ingesting to establish somehow, but you are not being fed by the carbon sources that you are eating. And in this case, it would be the same. Perhaps we don't want to directly enrich the soil with carbon. We just want to give those microbes an opportunity to establish and get them going.
Excellent. Yes. I have experience. We do compost amendment experiments, and it looks like there is some very different effects by adding the compost extract versus the amendments, and also the compost amendments have some kind of whole system effects on soil moisture, and also delivering a different form of N than maybe what weedy plants might take up. And so it might actually be a form of nitrogen that kind of is retained in the system a little bit more than just a nitrogen fertilization that oftentimes it might not be the best thing to do in systems, but you do need something to jumpstart plant recovery. So so there is a lot of subtleties between what compost does, but certainly something to look into, I think, especially when people are with a system that have grazers right in the system and trying to make those kind of more complex loops or reestablish those. Jim has a question that I think is kind of the elephant in the room about, so if we're emphasizing that this soil system is so complex, so diverse, and so important because of its complexity, is there any way that we can start thinking about actually complexity as the goal in restoration, and trying to think about that as the emergent property that we want to achieve in terms of, as Raul said, like a self-sustaining, right, system. So any thoughts about that, whether instead of focusing on perhaps one process, kind of turning towards complexity in terms of policy or monitoring or just our practice?
Well, I've spent more than 20 years studying soil heterogeneity and restoration, its role in restoration. So I think in theory, complexity is something that should be promoted because diversity begets diversity, right? So complexity and soil structure and nutrients and plant communities will all result theoretically in more niche availability, more niche dimensionality, and we're talking for all organisms, right? Soil microbes as well as the fauna and the plants and the insects, and and all other animals in the system. So I think that's a great point. I'm glad it was made.
I think that from everyone's other comments, I think it's absolutely probably shared by all panelists. How we actually get there might be a good 10-year goal, right? And so thinking about more of the system and really trying to take into account heterogeneity and complexity across different organisms. Jasper?
Yeah, I think I agree with it in broad terms. But just to bring up another point, I mean, a lot of research is focused on increasing diversity in soil microbes, for instance. But I think we're also realizing that it's much more about composition rather than species diversity per se. And so I think in general terms, it may be good to focus on complexity and to increase it, but you may be able to be much more effective if you know what components of biodiversity, what AMF are you needing in that system, what faunal decomposers are effective there, rather than simply saying, okay, we just need more of everything. So I think we if we zoom in on that, I think we can do a better job.
Excellent, yes. Getting a couple questions that are pushing us to think maybe across systems a little bit. And so I'd like to address that a little bit. So do you think that these dynamics, let's talk about some of the systems that we haven't talked about before. So maybe a little bit more tropical forests or deserts or wetlands, like how much can we kind of think about these general processes across the board? Or are there some systems where soil biodiversity is critical for restoration and maybe some that maybe it's not as much of a key consideration, or anything kind of at that kind of broad scale across systems? Any thoughts? Raul?
Yeah, I would say that as long as the immunity of the plants depend on soil microbes, that's always going to be a critical point. No, I mean, even though maybe in wetlands we don't have the earthworms or other big animals, I mean soil animals, but still the plants are always going to need the microbes for their health. So they're always going to be critical.
It's always important. I've always found it important.
Yeah, I agree. I mean, at some level, it is important. You need the microbes to cycle the nutrients. I guess if you keep, if they are not there, that will not happen. But again, I think, it's specific components of biodiversity that you need for more detailed things. And it's interesting to think about this, how indeed different, tropical to peatland systems in the tundra would differ in this. I don't really know if for instance, inoculation would generally be more important to do in tropical forests or in peatlands or in temperate grasslands. I mean, obviously there's a huge research gap in those tropical areas. So I guess that's a shout out to doing more research there.
I think it would be very helpful to have some of those kind of thoughts about just those general system dynamics and where and why in these different systems. So Susana, do you have any thoughts about that?
Well, of course, I agree that it is important in all systems, but it is true that there is a big knowledge gap in many of these different ecosystems. There are some things done in tropical areas more related to agricultural soils, for example, intercropping, or to improve the composition of soil communities in terms of suppressing pests and things like that, which I think is very interesting as well, even if it's not really restoration, but it goes in the same direction. Also, the tropical systems especially wet tropical systems, are very complex, very rich, also in terms of soil microbes. And I really wonder how they will behave with inoculations and with soil inoculation, if that will work, or the system will be so resilient that you cannot put anything different in there. But yeah, I mean, you always have to take into account the soil, otherwise plants are not going to be happy. So it depends on the particularities of the system and knowing what is important there.
Great. So we have about 5 minutes left. And so I just want to wrap it up a little bit. And, you know, we've talked about some really amazing things today. It's super exciting, kind of new approaches and new ways of thinking, and also some kind of frustrations or research gaps where we really want to move forward. So I'd like to end by asking, you know, what keeps you going and what really inspires you to be able to do this work, but also to transfer it to, you know, kind of change the mindset, whether it's land managers or policymakers or society as a whole, you know, in terms of transferring, you know, these this kind of very exciting system, but also really challenging issues to the greater good. I'd just like to hear kind of those end thoughts from all of you.
Well, I can start if you want. As you say, doing research on soil can be very frustrating, and and sometimes I have thought many times to change subjects and study something that you can see more easily. But then anytime that I do an experiment and see how important soil is and how different soils can change the whole result of the experiment of this or the study, and then I realized that it is very relevant to pass the message. And so still in many occasions, soil is used like a substrate, like a black box. That cannot be the future, because you cannot understand what is happening without understanding what is in the soil and what the soil is doing to the rest of the above ground organisms. I don't have much experience with managers or policy, so I'm not sure how that can be addressed, but I'm very happy to see that there are big initiatives also at the European level to address this and to get things in the right direction.
Yes, there are definitely some really exciting initiatives. Jasper?
Yeah, I think I will echo Susana's words. I mean, for me, the inspiration is mainly two things. One is the complexity of the challenge. So soil biodiversity is complex, the interactions are complex. That also makes it a major intellectual challenge, which I think motivates me, you know, to try and untangle that and see what influences what and why. And the other is the increasing attention that soil biodiversity is getting, and the importance that we have been increasingly able to show it has for soil functioning and also in restoration, and that's now, you know, that the public is very interested in this, the policy makers are very interested in this. I think that's very motivating. So there's a lot to do, but there's also a lot of people wanting us to do it.
Sara?
So I think there are two major motivating reasons for my research and what I do. And one and how it began was that I wanted to help inform practice so that restoration practices are more effective and informed. The second thing that keeps me going are the surprises. I mean, I really enjoy working in soil where, again, you can't see what's happening, but you collect according to a design within a system, and you are seeing these patterns over time in the data that you can't see. You can't walk out onto the land and see the phospholipid fatty acid biomarkers. You can't, well, you can kind of see the aggregate structure if you get in there. And then the surprises from ecosystem comparisons, right? When we compared the sandy and clay soil, I didn't expect nothing to be happening in those sandy soils. They had just as much above ground net primary production. The plants were there, the roots were there, right? And so that was a huge surprise. And then there is an important message for land use and policy, and that is some systems maybe deserve a little more protection than others if they can't recover, you know, using restoration practices, even within the time frame of programs and incentives to help.
That's awesome. Yes, I totally agree. The other thing is, it just seems like stakeholders and people that work the land really appreciate the soils too, right? And so they, it is a common currency that people really know if they have a long-term relationship to a particular parcel of land, how important the soils are. Raul?
Okay, yeah, I align very much with Sara. I also have two main reasons. The first one would be to promote the well-being and the health of complex living systems like us and ecosystems. And the other one is simply to understand the world around me, and soils happen to play a critical role in it. So it's somehow circumstantial, but here I am. It couldn't be otherwise.
Excellent. Yes. Yes. That's a great great last comment. So I would like to thank all panelists. It's been a pleasure talking to you all, and just really enjoy all your insights and research. So it's an honor. And then thank you to everyone who's listened and asked a question. I'm sorry that we couldn't get to all questions, but I hope we did justice. You can contact each of the speakers if you have a specific question for them. I'm sure they would be happy to answer. Also, you can follow follow it at the GSBI if you want to hear more about this. And then also for specific news about the UN decade, they're using hashtag Generation Restoration. And so that might be something just to look at and stuff, because there is a lot of exciting things then. So just want to thank you. I guess I'll transfer it over to you, Monica, to wrap it up if needed. But thank you, everyone. Really appreciate the last hour and a half.
Thank you, everyone. We would just like to say it once again, we are going on hiatus for the next 2 months. Please go to globalsoilbiodiversity.org for our new schedule in September. Thank you.